Hello, my name is Joshua Scott, and I am here to present my undergraduate research over the analysis of the protein-to-protein interaction of IpaC and IpaD in a bacterial virulence secretion system by fluorescence spectroscopy. Before I get started, I would love to thank Megan, Pallavi, and Dr. de Guzman. I really would not be here without them, so thank you so much for them. All right, so before I get started, I just want to give some background on the Type III Secretion System. Basically, this is just a tool used by gram-negative bacteria to inject their virulence proteins or poison into human cells. So let's look at this cartoon. It's very simple, and it's honestly gets the point across pretty well. The Type III Secretion System is just a protein needle expressed by gram-negative bacteria that gets injected right into our host cell. And you see in this very low resolution electron micrograph picture that the structure resembles definitely a protein needle. And we look at this animation and it shows it even further. So some notable diseases that come from this are food poisoning and even the bubonic plague. And it's also the #1 cause of death among cystic fibrosis patients because they get infected, they get this infection inside their lungs, and it just wreaks havoc, and unfortunately it kills them. All right, so back in 1998, we obtained a very low resolution picture of the Type III secretion system. You can see a base, you can see the needle, but as we progress, we start to develop a more defined image. But the questions are still uncertain of what is this piece and what is going on and what is this atomic structure. So this, as we progress from 1998 to 2019, it kind of leads us to our final endeavor, basically, is to figure out our goal in life, as Dr. Guzman will say, the translocon piece. We want to figure out the atomic structure in order to gain a better perspective on this system's function. So some awesome results came out in 2021 that gave us a better visual image of the tip protein. And this complex right here is created by 5 identical protein sets that interact together to form this tip. And what the tip does is just connect to the translocon piece. So we're seeing interactions between 1 and 2, 3 and 4, 4 and 5. But there's a weird gap here. And that gap leads us to what we want to find in our lab. So this gap kind of leads us to some possibilities with our research. We hypothesized that this N terminal right here on the IpaC translocon piece, remember this is the membrane piece, is somehow interacting within this gap, that gap that was weirdly there during that 2021 study. So over our lab, we are going to focus on that specific possibility. All right, we got our problem. We know kind of the end result we want. So how are we going to get there? And that's through just this general workflow presented on the right. For one, we're going to engineer some cysteine mutations into the protein. That allows us to input the dye we need to go through FRET. So after we get those cysteine mutations inputted, we can utilize E. coli bacteria and practically hijack their protein expression machinery to create a recombinant protein. Once we get that recombinant protein created with inside the E. coli cell, we isolate them, purify the protein, and concentrate the protein. And then, like I said before, we input the fluorophore dyes on that cysteine mutation so we can conduct FRET. Creating a visual for the protein interactions is vital for our lab. It's the end goal, it's what we need, and it occurs through FRET. FRET sends light into our labeled protein, it excites it to a higher molecular orbital. This energy then hopefully gets transferred to our acceptor protein, and that acceptor protein gets excited, and then once it relaxes to a lower molecular orbital, it emits a photon. And all of this gets picked up by our FRET machine, and from there we can create a spectrum and gauge if there's protein interaction or not. Just to reiterate what's going on within FRET, we have labeled proteins. So IpaC is labeled by PM, which is this structure right here. Light goes in, it excites this conjugated system, it goes to a higher molecular orbital, and then it kind of relaxes a little bit, and there's an energy transfer to our acceptor. Okay, our acceptor picks up within this conjugated system on the IpaD protein, and then it emits that photon, and that photon gets picked up by FRET. All right, so we understand the process and the procedure. So here's just another visual of our IpaD cysteine mutants. We got a mutation here, here, and here. All right, in order to create our recombinant DNA, we have to grow E. coli at the most optimal range so they can produce it. And that occurred at 4 hours of induced IPDG growth at 37 degrees Celsius, and after an overnight induced IPDG growth at 15 degrees Celsius. IPDG is just simply a molecule that helps us express our protein of interest. Our results showed that lane 1 had the best protein expression because it had the most intense band compared to lane 2. Lane 3 is just simply our control without any induction. So moving forward, this mutant can go through large-scale protein expression, induced growth at 47 degrees Celsius for 4 hours, and then it will be put through nickel chromatography. The same test went through with the other IpaD mutants, and the results showed that lane 3, which was the overnight induction at 15 degrees Celsius, was best for this particular mutant. And then in this mutant, the S173C, we saw no induced expression, and therefore that was the end of the road for that mutant. The protein purification results showed in lane 8 and lane 9, I got protein in the elution cycles. So that's our location of the protein. After protein purification of IpaD, I dialyzed E1 and E2 fractions, and that just simply creates a better environment for labeling. After dialysis, I concentrated using Omicron, which is this guy right here, and then I labeled. The final IpaD mutant went through a very similar protein purification process. However, the results were different. Lane 4, lane 8, lane 9, and lane 10 had our protein in it. Lane 8 and 9 saw dimerization. The best way to get rid of that would have been to add more DTTT, but that didn't really affect our experiment, so we just kept going with the process. IpaC goes through a slightly different purification process because it expresses as an insoluble inclusion body. So I purified it by dissolving IpaC in urea, followed by dialysis to remove urea. And then I also added a supernatant flow through in the purification. So zooming in on our results, lane 2 showed our test for expression. That was just overnight. And then lane 4 and lane 6 have our purified IpaC protein. Since we're studying a transmembrane environment, we have to replicate that. And the best way to do that is to study FRET interactions through micelle. Unfortunately, E. coli cell are way too complex to work with, so we use DDM as our artificial micelle. And we went through with FRET without and with micelle. All right, so we got our protein. Now it's time to get our results, and here are the official FRET results. But before I diverge, let's get used to some terminology. Control just simply means the individual protein FRET analysis, which is we're putting 150 microliters of IpaC protein into a cuvette and getting a FRET reading, and then we do the same thing with IpaD, put it into a cuvette, get a FRET reading, and then that spectrum is combined together in an Excel sheet to produce this control spectrum, this control plot. And the FRET is the experimental interaction. So IpaC and IpaD combine in a cuvette where a FRET instrument reads it, and we get a red line like this to gauge our actual experimental interaction analysis. So for a positive interaction to be justified, the peak on the acceptor region has to be above the control, and the peak on the donor region has to be below the control. So kind of like this, but not like this. So this right here altogether is a negative interaction, but in the micelle, we see another negative interaction. All right, so we got negative interaction results on the previous fret. Let's look at these. This one, we see the acceptor spectrum that we like. This means, okay, there could be some interaction, but within the donor spectrum, we do not see what we need. That experimental is just above the control. So that's a negative result altogether. And now in the micelle, again, this donor region is slightly below. That's what we want to see. But in the acceptor region, it is slightly below too. So now that confirms a negative result. All right, here are some more fret interactions. And again, we see a negative result. The donor peak is above our control, so that's a negative. And then our acceptor peak is below, so that's a negative. So negative overall. And then in the micelle, similar concept, another negative result. Okay, so I saved the best one for last. This is the closest results to a positive interaction I got. And what we're seeing here is a very close relationship in the experimental fret without a micelle. This is technically negative because it's slightly above. And then we also see a negative result here. So it's a negative interaction, but it's close. And then, so let's put it in a micelle. Let's see what happens. And we're actually seeing some positive fret here because it's just slightly below it. And we're seeing a huge intensity jump. So this is potentially a positive interaction. All right, so I say potentially because we need to go through more confirmation and ensure that those results are accurate. So I like it. If I had to say we had something, I definitely would, but more work needs to be done. Moving forward, I want to change the donor and acceptor concentrations. So we started with 1 micromolar of donor and then 1.2 micromolar of acceptor. And I just want to shift that and see if it, creates a better spectrum. So along with the Shigella system, I want to continue to keep working on that. And then I'm also interested in moving towards the Salmonella secretion system. Over winter break, I created some proteins and I just want to see how they interact with each other. So hopefully that's where it goes. Anyway, thanks for listening and hopefully you learned a little bit more about the Type III secretion system.